switch gears a little bit because I'm going to. I feel a little out of place here, but excited and privileged to be here to visit with you all today. I am going to be discussing the impacts of regenerative ag on both water storage and soil health. And so my role as an associate professor with both Texas A&M AgriLife Research and Texas Tech University is to evaluate the impacts of soil health management practices. At least a major component of that. We also do fertility work, but today I'm going to be visiting with you about some of the conservation agriculture that we're conducting that's aimed at both conserving soil as well as groundwater. So before we get started, I really need to set the stage. What is soil health? Soil health is the combination of biological, chemical, and physical soil parameters. We have to look at all three of those in order to really evaluate the health of the soil. NRCS defines it as the continued capacity of a soil to function as a vital living ecosystem that sustains plants, animals, as well as humans. And we look at this both from an inherent and dynamic soil properties perspective. So not just the inherent soil textural, which we just heard quite a bit about, but also the parameters that are changing with that management practice that's being implemented. So the NRCS defines management strategies as those that disturb the soil less, keep the soil covered as much as possible, keep a living root throughout the year, as well as diversifying the soil biota with both plant diversity that then has an impact on the microbiological activity of the soil. So one of the things that I was asked to speak about today is regenerative ag. Well, there's not a clear definition of what regenerative ag is currently amongst soil scientists. And so we tend to use the NRCS soil management strategies that include things like cover cropping, crop rotations, reduced tillage as our means of regenerative ag practices. And we do have a project currently that I'm going to talk a little bit about at the end of this presentation that is aimed at trying to identify a definition for regenerative ag, particularly in semi-arid environments. My research program, so I don't know if I mentioned, I'm located in Lubbock, Texas, so high plains area of Texas, semi-arid environment. I'll go through some of the details regarding the area here in a bit, but the idea or the purpose of my research program is to not only demonstrate but also quantify the impacts of conservation management practices on the soil chemical, physical, and biological products. So essentially we're looking at how these management practices that farmers are implementing, how those are changing these different properties of the soil, which then can have a positive impact potentially on both water conservation and soil conservation. So the high plains of Texas. On average, we're anywhere between 55 and 63, that should be degrees Fahrenheit, not Celsius, sorry about that. We receive anywhere from 16 to 22 inches of rainfall annually. Of course, this year, we all know across the state of Texas, that's definitely not going to be the case unless this rainfall continues that we are receiving. Average wind speeds are 12.3 miles per hour, and we have approximately 195 to 255 frost-free days. So what does all this mean? It makes it really challenging to try to grow agricultural crops. And one of the main reasons for that is that our potential evapotranspiration can exceed our rainfall from anywhere to 2 to 3 times as much. So which is one of the reasons that about 50% of our production acres rely on irrigation. That irrigation is being sourced from the Ogallala Aquifer. One of the long-term sites that we work in is located in the southern portion of the High Plains, and this is in Lamesa, Texas. It's about 70 miles south of Lubbock. At this location, we have a research farm that we call AgCares. It's the Agricultural Complex for Advanced Research and Extension Systems. The soil type at this location is an Amarillo fine sandy loam. We do have limited irrigation capacity at this site. In my mind, this is the perfect site to represent the most harsh environment that we're trying to grow crops in the southern high plains of Texas. Sandy soils, limited irrigation capacity, and we're trying to grow a cotton crop in this environment. So of course, we're going to see those changes from implementing conservation practices, which I'll get into here in a second. We had a project several years ago with the NRCS to evaluate the impacts of these systems, but also compare it back to a native system. Because ideally, we would want to farm as nature happens on a daily basis. And so we wanted to evaluate the soil health properties of these native systems compared to our managed agricultural systems. At the native site, according to NRCS records, had never been plowed, at least for the 80 years that they had on record. So it could have been longer than that, but 80 years was as far back as their records went. Both locations, again, are in Amarillo fine sandy loam. This is a benchmark soil series for the Texas Southern High Plains. About 50% of our agricultural acres are in Amarillo fine sandy loam. The primary uses are going to be ag production and rangeland. What's unique about the soil is it's about 80% sand. That's pretty tough when it comes to trying to grow a crop because of the lower cation exchange capacity, the decreased water holding capacity, and so again, a very harsh environment to try to work in. So we have 3 different systems that we're evaluating, 2 of which were established in 1998. Of course, we have our business as usual practice, which is conventional tillage, winter fallow, a continuous cotton cropping system. We have a no-tillage system with a rye cover crop. Again, both of those were established in 1998. And the data that I'm going to be presenting to you today was 20 years after the establishment of these systems. So definitely a good representation of what happens from a long-term perspective of implementing these systems. And then in 2014, we implemented into the existing rye plots different cover crop treatments. The cover crops that we were looking at, it was a mixed system of 50% rye, 33% Austrian winter pea, 10% hairy vetch, and 7% radish. And again, established in 2014 in what had previously been the rye cover crop no-tillage systems. And just to look at what a traditional system looks like compared to one where we are implementing winter cover crops. So a traditional cotton cropping system in the high plains of Texas would be fallow from about November, from November until May. Our cotton growing season is going to be mid-May until about November when we harvest. So we're trying to get a cover crop established in the middle of November, early December, then we terminate it in mid to late March, possibly early April, depending on the season, and then we may have a 4 to 6 week window where it just sits idle before we plant the next cotton crop. This is really challenging. So from a cover crop perspective, we're trying to get something planted in November. We have to deal with freezes, things that make it really challenging. We don't necessarily get active cover crop growth until sometime in February. So we have a very short period to get much biomass produced that's going to help in keeping wind erosion from occurring, improving water infiltration like we just heard about. And so it can be a challenge depending on what environmental conditions that are occurring. And that's evident here. So here we're looking at cover crop biomass in pounds per acre. And here on the left, we have the no-till mixed species. And then on the right, it's the no-till rye from 2015 through 2021. And you can see it's extremely variable. and that 100% goes back to the environmental conditions or the weather that we experienced within a given growing season. So reduced rainfall can reduce the amount of biomass that's produced. If we have a colder winter, we're going to have less biomass produced. But the reason that it's important for us to acknowledge not only the variability, but also the amount of cover crop biomass that's being produced, is it's directly impacting the changes that we see in soil health. So we were just hearing about different soil amendments. One of the best things that we can add to a soil to increase the cation exchange capacity, to enhance the microbial activity, is going to be crop residues or plant biomass. And that is what is directly impacting the health of the soil. Some of the major parameters that we look at from a biological perspective when it comes to soil health is soil organic carbon. That is probably the overlying greatest contributor to the microbial activity that's occurring in the soil. So we look at soil organic carbon, we look at total phospholipid fatty acids, which is an estimate of microbial biomass, and then we look at enzyme activity. Enzyme activity can represent the carbon cycling and the nitrogen cycling that's occurring in the soil. So here, the figure on the top left, we're looking at two different depths, 0 to 5 centimeters and 5 to 10 centimeters within the soil profile. And then we have our 4 different management practices: conventional tillage, rye, no tillage, mixed species, no tillage, and then remember our native system that we're using as a comparison here. What is really interesting about these systems is that where we have a managed system, where we're implementing a cover crop and reduced tillage, we're increasing our organic carbon levels, we're increasing our microbial biomass, as well as our enzyme activity. The 2 figures on the bottom are looking at beta-glucosidase and beta-glucosaminidase, which are carbon and nitrogen cycling enzymes respectively. And in both cases, where we have a cover crop implemented, because of that added residue, we're increasing organic carbon and we're increasing the microbial activity in the soil. For some reason, my figure here is not showing up. So one of the things, of course, is that we then look at the relationship to crop yield. A farmer cannot stay in production if he's not also maintaining his yield. So we want to evaluate how yield is changing compared to the positive benefits we're seeing of implementing cover crops into the system. And unfortunately, we do not see a relationship between crop yield and soil health. We actually see quite a bit of variability, just like we saw with the cover crop biomass. So again, we're enhancing all of these biological properties of the soil, but there's something still limiting the system because we don't see a consistent benefit to the cover crop in the improved soil health to cotton production. Here we're looking at cotton lint yield in pounds per acre from 2015 through 2020. The gray bar is our conventional till system. The green is no-till with a rye cover, and blue is no-till with a mixed species cover. The only 2 years where we saw significant differences between these systems was in 2016 and 2017, and unfortunately, the conventional till outperformed or produced greater lint than the rye no-till system. And so that leaves us questioning what is going on. And if you talk to any farmer, if you talk to any scientist that has been working in the high plains for long periods of time, they're going to tell you it's water. We're growing a crop during the winter when normally we would be capturing and storing any sort of winter precipitation that occurs. And when we grow a crop, that crop is going to utilize any moisture that may be in the soil. And so, of course, one of the things that we look at is soil water. Here we're looking at water use efficiency from 2015 through 2020 and our 3 different systems. There are no differences in water use efficiency between these 3 systems. So then the next thing that we want to look at is the actual soil moisture. And this is where it gets really exciting and really interesting. So here it's soil moisture in inches from a 0 to 60 inch depth in our different growing seasons that I've been discussing. Same color scheme with conventional till being the gray bar and no-till rye blue, no-till mixed species in green. And then we also have our different time points pointed out here. The cover crop termination, which is a chemical termination, occurs sometime in March or April, and then we plant our cotton crop 4 to 6 weeks following that. But what you'll see consistently is that there is going to be this decrease in soil moisture prior to terminating the cover crop. So like everyone says, we do see a slight deficit in stored soil moisture to a 60-inch depth where we have a cover crop planted. But what happens after we terminate that cover crop? We have greater soil moisture, which makes sense. It's increasing infiltration, increasing capture of any sort of precipitation or irrigation event that happens. And so throughout the cotton growing season, we actually have greater stored soil moisture compared to the conventional till system. So based on this data, we're able to rule out the fact that it's not soil moisture that is limiting cotton lint yield. There's something else within the system that is limiting production of that cotton crop. So I have a colleague, and he has been doing this for years and years and years, and he's like, well, what if there's a specific depth? So this is cumulative soil moisture. We're going out with a neutron probe and we're measuring the soil moisture every 20 centimeters. And so we're able to look at soil moisture by those individual depth increments. And so it could be a possibility that we have a specific depth where we do see a deficit that's not being made up throughout the growing season. And so the next stage was to look at that by individual depth. The top figure is our conventional till system, business as usual. The lower figure is no tillage with a rye cover crop, where we have the darker colors, blue, that's going to indicate greater moisture present. The orange and the red is going to be where water is more limited, a lower volumetric water content. And just like the cumulative data indicates, there is no depth where we're seeing less moisture in the no-till system with the cover crop compared to the conventional till system. So from the perspective of stored soil moisture, the cover crop is benefiting our water dynamics in the system compared to the conventional tillage. So then the next thing that we want to look at is overcoming the yield reduction by altering the nitrogen management strategies that we're using. So we looked at nitrogen fertilization at different timings. So I have 5 minutes left. I'll try to get through this. So we have our rye cover crop again, a cotton wheat rotation, and continuous cotton. The idea behind this is altering what our extension recommendations have been for the last 20 to 30 years by moving our nitrogen earlier in the season. So traditional recommendations are to space it out throughout the season, putting a larger amount further into the season. And what we found is, especially where we have a cover crop in place, that if we move our nitrogen to earlier in the season, in the case of the pre-plant nitrogen application or the early emergence application, we increase our lint yields by 16 to 20% compared back to the conventional business as usual system. Last few slides that I want to get through is looking more specifically at carbon. Again, I know we're at a groundwater summit, but carbon is one of the big discussions that is going on right now, not just in agriculture, but across different disciplines. And it does have a big impact on the water storage of our soil and conservation of water. So we have 3 different farms that we've been working in. It's really nice because it shows a gradient of soil textures and management strategies and irrigation capacities as well. Because as you'll see here in just a bit, irrigation capacity really does influence what we're seeing from a carbon storage perspective. The first farm is north of Lubbock. It is a Pullman clay loam, only 20% sand and 30% clay. We have 3 different systems that we're looking at here: our business as usual, our cotton wheat rotation, and our continuous cotton with a cover crop. And what we have found in this heavier textured soil, more clay, is that a rotation is going to be more beneficial from a carbon storage perspective compared to the cover crop system. The best way to look at these systems is looking at the influence of the conservation system relative to the conventional till system. And the figure on the right is illustrating that. Here we have the change in organic carbon compared to the conventional till system in tons per acre. So where we have a rotation under the higher irrigation level over this 7-year period, we've seen an increase in organic carbon to a 36-inch depth by about 3.5 tons per acre. So over that 7-year period, we've seen that increase due to that management strategy. With a cover crop, it's less than 1 ton per acre. And then when it comes to the lower irrigation level, we're seeing less organic carbon than we are even with our conventional till system. The next system is being conducted at the Lubbock Research Center. It was established in 2015. It's an A-cuff loam, so again, that textural gradient. Here we are able to look at CO2 emissions. The one thing that I want to point out on this slide that often comes up in conversation is how does a cover crop influence carbon dioxide losses from the soil surface? And here, where we have the maroon bars is our no-till cover crop system. And if you were just to look at the gross losses of CO2 from these systems, there's actually greater amounts of carbon dioxide being lost from the soil, which makes sense. Again, you have a crop growing more throughout the growing season compared to the other systems. But when you look at the actual net carbon flux, it is less than the conventional till system. And on average, we decrease CO2 emissions with the implementation of a cover crop into that system, which results in greater organic carbon compared to both our no-till system and conventional till system. This is really important. We talk in here within the agricultural community that just reducing tillage might be the answer to increasing carbon storage in our systems. But we have to have that added residue from a cover crop or from a crop rotation, because where we have the cover crop compared to just the no-till system, we have greater organic carbon present compared to just the no-till system. Last one is again taking you back south of Lubbock to the Lamesa location in that sandy soil. Here we have organic carbon levels where we have a cover crop and no tillage. We have greater organic carbon, not only profile organic carbon, but also at individual depths, we're seeing an increase compared to the conventional till system. We have a study that we've been conducting with the Texas corn producers on farmers' fields to evaluate organic carbon levels. These were established in 2017. Soil samples were collected in 2020. And we have seen absolutely no change in that short 3-year period. This is something that takes time. It's not a quick process. Most of the data where we are seeing those changes, it's at least 5 to 10 years before we start seeing any benefits to these systems. So in summary, again, we see greater water infiltration, water capture, water storage with the implementation of regenerative practices. We see improvements in overall soil health. Water is a major component of soil health. If we don't have the water, we don't have the biological activity. And so all of this really does tie together. We see a reduction in erosion, but we have to figure out what's going on from an actual productivity perspective of these systems, because without the production, the farmers aren't going to be able to continue to implement these practices on their farm. I'm going to go ahead and skip to the end here. We do have several other projects that we're conducting. One of those is in collaboration with the Texas Water Resources Institute out of College Station, where we are, again, trying to define what regenerative agriculture is across semi-arid environments. We're working across the southern portion of the Great Plains, both in Texas as well as in Oklahoma, and looking at all these different environmental parameters that I've discussed, but on a much wider scale than what I've discussed with you today. So again, I'm going to skip through this. I don't like doing this, but I'm feeling a little pressure over here. So, so thank you very much again for the invitation to be here.